Good evening, ladies and gentlemen. Welcome to BrisScience, the University of Queensland's free public lecture series on science, held once a month here at The Edge, part of the State Library of Queensland. We're, of course, enthusiastically presented by the University of Queensland, and we really appreciate The Edge hosting us here every month. It's a fantastic venue, and they have lots of great things you should check out. And I am your MC for this evening, Joel Gilmore. I'd also like to acknowledge the traditional owners of the land on which we're meeting tonight and pay my respect to elders both past and present. I also want to recognise those whose ongoing efforts to protect and promote Aboriginal and Torres Strait Islander culture leaves a lasting legacy for future elders and leaders. Tonight we have, of course, a fantastic speaker. BrisScience is all about bringing not just the best scientists, but also the best communicators to share their research. And I will introduce Melissa in just a moment. After her presentation, we will be taking questions. And here at BrisScience, we take questions by question slips, which you might have picked up on your way in, or you can write them down on anything you want. And after the talk, we'll come around and collect those and get through as many of them as we can before the night is over. And then we have some food and drink afterwards, and we'd love you to come and join us and continue this discussion afterwards. We also take questions over Twitter, so you can use hashtag BrisScience, but please, if you've got your phone here, if you could set it to silent now, that would be wonderful. Otherwise, we'll all get to do the point and shame thing afterwards, which we don't want to do. And I think that's all of the housekeeping for now. So it is my great pleasure tonight then to be able to welcome Professor Melissa Brown from the University of Queensland. She is a leading Australian researcher, particularly in the field of breast cancer genetics, including understanding how human genes produce tumour suppressor proteins even, and how that can go wrong. And that's what she'll be talking about tonight. Melissa is also now, this year, the new Executive Dean in the Faculty of Science at the University of Queensland and is a strong supporter of women in STEM. So could you please join me now in welcoming Professor Melissa Brown.
When I was young, I used to wonder about the future. I used to wonder which pathway I should take and where it would lead. I wondered what I'd be good at, I wondered what I'd enjoy, and I wondered how I could ensure that I had a long and healthy life. I often thought that it would be helpful to have an instruction book. Looking back now, if I had had an instruction book, I would have done things very differently. I wouldn't, for example, knowing what I know now, have chosen to spend my days like this, on the bayside beaches of Melbourne. Knowing my genetic makeup, it was not a smart idea to try and be like sunburnt Barbie. I didn't realise, because I didn't have an instruction booklet, the chaos that would cause me some 30 years later. Thanks to research over the last 30 years, everything has changed for the next generation. Because of the research that's been undertaken, we now have a very small, and I'll emphasise very small, and I'll come back to this, glimpse into our instruction booklet. This glimpse has enabled the next generation to see what's in their instruction book and to make some more informed decisions about their future. The example I'm going to talk to you about is Angelina Jolie, who, as you know, is a very famous actress amongst other things. Angelina had a strong family history of breast cancer, but she didn't know whether or not she had inherited the defective gene that causes an increased risk of this disease. Because of the amazing research over the last 30 years, which I've had the incredible privilege of witnessing and the wonderful opportunity to play a very small part, it was possible for Angelina to take a genetic test and from that to know that she had in fact inherited the defective version of one of the breast cancer genes. Knowing that meant that she knew that she had a significantly elevated lifetime risk of breast cancer, over 70% chance that she would get breast cancer in her lifetime. She therefore chose to make the informed decision to minimise the risk by removing all of her breast tissue through a double mastectomy. So the question arises, how did we get to the point that these tests were available? How do we really know what's in that instruction booklet and what is the future in this area? So what I'm proposing to do is take you on a journey. I'm going to turn this room into a magical shrinking machine which is going to take you inside the cells and the DNA of Angelina. I'm going to focus on a skin cell because most cells have a complement of DNA, and for most cells the DNA is essentially the same. So what we're going to do is go inside one of these cells. So Todd, if I could have the lights down, because we're in a spaceship, I want you all to hold on to your seats as we go into the incredible shrinking machine and we go inside one of these cells. So as we get closer, this is what we can see. So these are skin cells, and they're essentially a bag of chemicals floating in water. They contain micro machines or nano machines that do all sorts of wonderful things, like produce energy and produce proteins. And then right in the middle, as you can see here, is something we call a nucleus, and this part of the cell is the home of our instruction booklet. So let's shrink ourselves down even more and go inside the nucleus, and this is what we see, these wonderful, wonderful structures. These are one of my favourite things. Ever since grade 5 at high school, I first saw chromosomes, that was it. Love at first sight. So we all have 23 pairs of chromosomes. We have pairs of chromosomes because we inherit one from our mother and one from our father. The chromosomes comprise DNA that is tightly wrapped and compacted with the help of proteins into these structures. And if we zoom in, we're going to shrink again and zoom in even further, we can see the structure of DNA. And you'll often see this around everywhere you drive around, and you often see this double helix structure on, advertising various different things, which is sort of interesting having lived through the journey. So DNA is a polymer, and a polymer means that it's made up of lots of different subunits, and so it's a bit like Lego, if you like, where you've got a whole lot of pieces of Lego all joined together. The subunits in the case of DNA make sure I push the right one now. These chemicals here, which are made up of carbon, hydrogen, oxygen and nitrogen, and they're called bases, but for the purpose of today, where we're talking about our instruction booklet, they're letters in this instruction booklet. Unlike our much bigger alphabet, the letters in our instruction booklet, there's just 4 of them, and they're referred to as A, G, C and T, which is a symbol for these different chemicals. They're all stuck together, and they're in an order which matters. The order of these letters matters, just like they do in an instruction booklet. And so what can happen sometimes is the DNA can get damaged. We are constantly barraged with damaging agents, whether it be chemicals or radiation or errors in copying the DNA, and that affects these letters. Sometimes we lose the letter, sometimes we change the letter, sometimes we duplicate the letter. But the point is, just like if it was a book that we were reading, if the letters start to change, all of a sudden it doesn't make sense anymore, and the purpose of the letters is no longer readable, and we don't get the right products of our genes. In the case of the breast cancer 1 gene and the defective version of which, so we all have a breast cancer 1 gene, males and females, everyone has one. For patients that have an increased risk of the disease and a strong family history, they sometimes, in a small proportion of cases, have a defect in that gene. Now, in the case of BRCA1, which was the gene involved in Angelina's case, the product of that gene is a DNA repair protein. So DNA repair proteins function to make sure that when this happens because of chemical or radiation damage, that repair can be, damage can be repaired. So it looks something like this. So again, here is our DNA double helix, and these blue and yellow shapes represent DNA repair proteins that are scanning along and fixing the DNA repair. So if you inherit a defective breast cancer gene, you don't produce these blue and yellow molecules, so that when there's damage, as we had on the previous slide, it doesn't get repaired. And the consequence of that, after much damage, is that it starts to accumulate, and you get something called genetic instability, where you've got mutations in multiple genes, and that increases over time. The result of that is defects in a number of different pathways, including cell division. So this is just an image now of 2 breast cells that are dividing, and they normally divide, but you get uncontrolled cell division. You get increased DNA damage and increased mutations. You get defective differentiation. A whole lot of things go wrong. Let me show you on the next, now we're fingers crossed in our spaceship that it's going to work. Yes, okay. So let me show you what I'm looking at here. So these are normal cells, the blue ones. Here, the green ones are cancer cells, and you can see that they're dividing more frequently than the blue cells. This just results in too many cells in the one place. Then what can happen is, because they've got nowhere to go, they start to expand and invade into nearby tissues. They need to survive, they need oxygen and nutrients, so they attract new blood vessels to that region. That's not a problem. What's a problem is when part of that tumour breaks off, and as you can see, it gets into the blood system, and then it can regenerate that tissue somewhere else in the body. In the case of breast cancer, if it moves to the brain, to the blood, or to bone, that is what becomes a major disease issue. Todd, we can have the lights back up now. We're back in Brisbane. Okay, so the question then arises, how do we go from this knowledge to where we are now? And that is because we know what the gene is, we know that it can cause these tumours, but how do we actually identify those individuals that carry that increased risk? Because we understand the letters specifically in that gene, we know what they should look like, and we now have the technology, and this is an important point, because the technology changes all the time in this field, and it's the improvements in technology which have really enabled us to do what we can do now. So because we know what the gene looks like, we know what happens when it goes wrong, for individuals that have a family history that looks something like this, we can help. So this is a pedigree, a classic pedigree of a BRCA1 family. So the circles represent females and the squares represent males. If there's a line through them, that means they are deceased. The individuals where it's coloured in, that means that they are affected individuals. So what you can see here is that the grandmother in the family was diagnosed with ovarian cancer at the age of 49. Her daughter and her granddaughter were also diagnosed with early onset cancer. Her other daughter had breast cancer when diagnosed at 42, and the individual that we're interested in here has a 50% chance of inheriting that defective allele, but she doesn't know whether or not she has it. As a consequence of research, products like this have come onto the market. Now, the gene was isolated in 1993, and shortly after, a variant of this product was developed and was available. But it's taken 24 years before this has now become a Medicare rebatable genetic test in Australia, and that's really just because the High Court, year before last, decided not to recognise the fact that Myriad Genetics, who had developed this test, was holding a patent on something that was naturally occurring. So this test is now available for those individuals with high risk. It's not a population screening tool. These mutations are generally only found in individuals with a very high risk, where they have early onset cancer in the family, and a number of individuals, as I showed you on the previous slide. The other outcome of research in the BRCA1 field has been the development of targeted therapeutics, and that's come from a great understanding of DNA damage repair pathways, and knowing that defects in those pathways are what underlies this disease. So it's been possible to develop these particular drugs here, which are PARP inhibitors, that specifically target and destroy cells that have a high level of BRCA1 associated DNA damage. These drugs have been through clinical trials and are now available in the market. So these are great outcomes. So the BRCA1 mutation is an example where a single gene mutation can cause a significant increase in risk of disease, even though there are other genes contributing to this process along the cancer pathway. There are many other examples of single gene diseases, as outlined here. So these are just a few examples: Huntington's disease, cystic fibrosis, sickle cell anemia, and various muscular dystrophies. Now, many of these diseases have been known about for a long period of time. Through a mad rush in the 1980s and 1990s, the genes that are defective in these individuals were isolated. And again, some really good news stories that's arisen out of this research. Let me give you a couple of examples. So in the case of muscular dystrophy, it's an X-linked disorder, so it affects men and boys. The gene was identified in the 1980s, and Steve Wilton, who's a friend and colleague who works at Murdoch University in Western Australia, has been really successful in developing a novel therapeutic against this disease. So what happens in this disease? It's a very large gene, so it's got lots of words in its paragraph, if you like, in the instruction booklet, and there's a common mutation which occurs in a large proportion of cases. He has developed an RNA-based treatment that can actually chop out, through an enzymatic reaction, to remove the affected word in that paragraph. So rather than mucking up the whole paragraph so it doesn't make sense and doesn't work anymore, by removing the defective word, the machinery can still make sense of that paragraph. So that's been currently going through clinical trials. The initial results are incredibly promising. I've seen some videos. I tried to find them. I couldn't find them on the web, but they're around, which showed boys like this being able to get up and run around a back garden. So that's really, really heartwarming, really fantastic. Another example is this one here. Now this is Alexis Beery and her twin brother, who had a neurological disorder when they were young, and it was characterised by not sleeping at all, and as a parent we can only imagine what that would be like. They had muscle weakness, constant vomiting, and were towards the end of the day were unable to stand up for long periods of time. The family went to doctor after doctor after doctor with no firm diagnosis. The diagnosis included colic in one case, cerebral palsy in another, but every diagnosis was wrong. Because of the technology, and because it is now possible to sequence entire human genomes, these twins were subject to DNA sequencing several years later to find the single gene that was involved in this particular disease. They both had inherited two defective parts of this gene, and because they knew what it did, it was involved in the synthesis of an important neurotransmitter that was missing in these children, they were able to treat them with a drug that specifically targeted that defect. And this is a recent picture of these individuals. Yeah, no, it's just spectacular, and it's spectacular, and it's just spectacular. Anyway, so not all genetic mutations are responsible for very, very serious diseases. There are many genetic changes that underlie more common and more complex diseases. So whilst there are examples of cardiovascular diseases that result from a single gene mutation like the ones I've been talking to you about, and whilst there are also examples of neurological diseases, the vast majority of these fall into the category of complex genetic diseases where there are multiple genes that are contributing to a particular phenotype or characteristic, and that's what makes it a little bit more, literally, a little bit more complex. So we can represent that relationship on a graph like this. So if we look at the muscular dystrophy example here, it's one gene, it's a severe mutation, and it's very rare in the population. There are examples, as I mentioned, of cardiovascular and neurological disorders that fall into that category that are due to mutations in single genes. However, for the majority of these, they sit more in the middle as shown here. So these are cardiovascular, neurological, and a number of autoimmune diseases where there are lots of genes contributing. The effect of each one is fairly moderate, and they are relatively common in the population. So these are these complex disorders. Now that is an example. I mentioned not sunbathing, for example. It's not that I have an inherited cancer gene or a syndrome, but my combination of genetic variants, of common variants that are in my genome are such that I'm much more sensitive than many other people are to sun damage of cells. Now, in addition, our characteristics are largely genetically determined, so they might include our personality and our looks. These are examples where they are common variants. They're mild, they have very little effect on the specific gene, and there are many genes contributing. As we move from here, of these single gene disorders, through to here, of these common genetic traits, the ability of the environment to modify the effect of that mutation increases significantly. So when we were talking about breast cancer down here, we were talking about something where really it was very, very high probability of getting the disease, and only drastic measures could be taken to modify it. As we move towards here, and we can see on the next slide, these sort of characteristics can be modified by a large number of things. So we can, Nicole doesn't look like that anymore, right? It's because we can modify her genotype. We can change the colour of people's eyes. Depending on how people are educated, no matter what their IQ, they can do differently at school, and personality can be modified by experience. So again, like the suntan Barbie, if you have a combination of very common and low-risk genes that are working together, you increase the chance that you can modify that risk. So let's return to the breast cancer example. And I've told you already, in the case of the BRCA1 gene, we're looking at variants that are severe single gene, and they're rare. Those variants are rare in the population. But breast cancer is not always like that. There are many, many other genes that can contribute to this disease, but they move up this spectrum right up to being relatively common and contributing in concert together to increase the risk of cancer. As they move up that spectrum, the number of genes that are required to cause a significant risk of breast cancer increases significantly. So whilst in the case of Angelina, there was a 7 to 10-fold increase in her lifetime risk of breast cancer, as we move up and we start to identify and characterise the genes up here, the risk that they confer is sometimes only 20% above the normal population risk. So the normal population risk of breast cancer in Australia is about 1 in 11, depending on which analysis you do, or let's say 1 in 10 to make it easy. In the case of Angelina, so rather than being a 10% risk as the normal population risk, she had a 70% risk. In the case of these people who have one of these variants, they'll have a 12% risk. So if the population risk is 10% and your personal risk is 12%, you're unlikely to make a decision to have a very severe clinical treatment. But if you know that if rather than having just that gene, that you have many genes that together can give you a 30% risk, 50% risk, 60% risk, then that changes the decision-making process. So recently, and I'm talking here, so this is, just to show you that we don't know where all of these genes are, so this is one of our challenges. But recently, and so this is a press release from the Queensland Institute of Medical Research, Berghofer, from the 24th of October this year, so just recently, describing publications that were published in the journals Nature and Nature Genetics. This study, which was co-led by Professor Georgia Chenevert-Trench, has analysed 275,000 women and been able to identify 72 new genetic variants, so they fall on that left-hand side of the graph, that in combination can cause a significant risk. So our genetic testing is now changing from looking for a single change in a single gene to looking at which combination of genes have we inherited and how might they work together or not. Some genes can be protective. So if you've got a balance of causative and protective genes, your population risk or your lifetime risk might be the same as the population risk. But if you are unlucky and you've inherited all of the detrimental versions of those genes, then you're in a more dire situation. One of the other challenges that we face is knowing what these variants do to the structure and function of our genes. In the case of the BRCA1 gene, 50% of the clinically identified variants fall into the category of being unclassified variants. And what that means is that we don't know what is happening in these patients. They can have the genetic test that Myriad Genetics developed, and they can come back that, yes, you have a variant in that gene, but in 50% of the cases, we don't know what that means. That causes a huge problem for medical doctors and for genetic counsellors, because we can't tell the patients what to do. We can't provide them with guidance. Now, that's just in the high-risk genes. When we start looking at the low-risk genes, we don't know what most of the variants do. So we've got a long way to go. To address this, another friend and colleague from QIMR, Amanda Spurdle, has set up an international consortium called Enigma. Now, Enigma is the evidence-based network for the interpretation of germline mutant alleles, and it focuses on breast cancer genes. And this consortium is a group of more than 50 laboratories, clinical testing laboratories and research laboratories from around the world that is tasked with looking at these clinically identified variants and trying to work out what they do. This is my favourite thing, and this is my favourite research project, and I'm just going to spend a couple of minutes telling you about this. So my research for decades now has been looking at the BRCA1 gene, and I am particularly interested in the parts of the BRCA1 gene that determine when it is switched on and producing its RNA and protein molecules, and when it is switched off. Specifically, I am interested in knowing which one of the variants, which regions and which variants might actually contribute to disease. So if we look at this image, we've seen chromosomes before. We know about our double-stranded helix. We can see all the different colours here, which represent the letters, and we can see this blown up here in the bottom. What happens when a gene is expressed is a molecule, which is shown here in yellow, comes along and binds to the DNA and says, ready, set, go, and starts producing an RNA copy of itself. This is a process called transcription. The regulation of transcription is incredibly complex and involves a whole lot of DNA elements and proteins. So DNA elements sit before the gene actually starts. They sit on other parts of the DNA that can sometimes be a long way away. They sit at the end of the gene as well. So the idea of our research project is to try and understand, one, where all those elements are, and two, what happens if a patient inherits a variant in those regions. Does it mean that the gene doesn't get switched on at all, in which case they would be at a similar risk of getting the disease as if there was a mutation which took out the function of the protein? Let me demonstrate this in another short video. So this video shows the process of transcription. I'm going to outline the regions that we're particularly interested in. So the red is the DNA double helix in this animation. The yellow proteins here are the RNA polymerase molecule that I showed you on the previous slide. This is the enzyme which produces the RNA. It's raring to go, as you can see, but it can't start until a whole lot of friends come along and bind to this region and says, yes, it's okay to start producing it. So here they are, and they've joined the party, and once it's ready, it now needs other proteins to join in and other regions, and I'm just going to show you up here in a minute because DNA loops around, and it's going to come around in a minute, just there. So that's some of the regions that we're interested in finding. Where do the proteins bind to the DNA that switches it on? Once it starts, it's off like a train. So here's the DNA double helix, and here's the protein machinery binding to it. All these popcorn-looking structures are the monomers in that polymer. So they're like the Lego blocks that I mentioned before. And they need to join up together in the right order. So they come in to this enzyme structure and they come out in the right order. And this is And this is predicted to be the speed that it actually happens in cells. It's amazing, it's fantastic. So this is what I'm particularly interested in. I'm interested in where do these proteins bind and what happens if those proteins are unable to bind and switch on expression. Now, I said at the very beginning that as a consequence of 30 years research, we now have a small, and I mean very small, glimpse into our instruction manual. When I say small, it's about 5%. So we only, despite all of this research, all of these billions and billions of dollars that have been spent, we only actually understand 5% of the human genome. And of that 5%, even in probably one of the best studied genes in all history, we don't understand what 50% of the clinically identified variants do. So this raises a number of questions if we go back to the future. And the questions are about how far we take this. How much do we actually want to know? If we don't understand most of the genes and most of the effects of the variants, is it useful information? If we don't have treatments available for many of the diseases, is it useful information? Is it risky to have too much information about these genes? Do we actually want to know our future? Do we want others to know our future? Or do we want to enjoy our life and the journey that it takes us on and see where it goes? I'm going to argue that we do want to know. We do want to know what the variants mean, and we do want to be able to have good treatments. But that's not going to happen in most cases tomorrow. It's probably going to be somebody else standing up here in 30 years' time telling you what we can now do thanks to technology. Thank you.
Thank you so much, Melissa. We'll give you a chance to catch your breath. We will take questions now, so if you have any questions, write them down on the question slip you picked up on the way in, or anything else, $10 note, $20 note, anything is fine. And we'll also take some questions over Twitter. And while you're doing that, and Leonie and Dominic will come around and collect those, so just sort of wave them in the air. While you're doing that, next month we have a fantastic talk on artificial intelligence. And this is including a couple of great researchers from QBI at UQ, including Professor Jeff Goodhill, who I've seen present several times and is just brilliant, he's a neuroscientist from the Queensland Brain Institute, talking about how brains work, about some of the recent work on AlphaGo that you might have heard about, the self-learning neural network there, and some other colleagues. So should be a fantastic evening, and make sure you book your tickets ahead of time for that. All right, so I'm going to go to start asking questions, if you're good.
I think so.
Great. So I'm going to go to Twitter first. If I can... Okay, so the first question is... sorry, I'll paraphrase because my phone is being slow. First question is, what are the implications for new gene editing technologies such as CRISPR for this and other areas?
Okay, so this is a great example where advances not only in our knowledge but also in our technology can change what we can do. CRISPR is a technique where you can actually change the genome using a process of genome editing. Now, there are all sorts of implications of this research. It's been tremendously helpful in trying to understand the function of various genes, in trying to understand the impact of variants in genes. What I worry about is that 95% split. We only understand a small part of the genome, and we don't really understand what else might happen when we use this editing technology. So I think we need to be cautious, and it's partly about experimental design, it's partly about taking care and interpretation of data, but we do need to be cautious because sometimes you get off-target effects where the editing technology can actually make changes elsewhere. You might get effects and make assumptions because you think what's happened is actually not what's happened. Look, I'm always conservative with these things. I think it's tremendously powerful and it is the way of the future, but it's early days.
So perhaps a related question to follow up on that is, why is it so hard to figure out what a gene does? I guess that's the 50% question, a 5% question.
Sure. Well, it's a question of how do you actually go about it. So let's say that we've got an individual who has had their genome scanned and they come back and they've got a change in one of the letters that says the letters have changed in the word, and if the word is part of the gene, and the gene is the whole paragraph, that you've changed some letters. So if it was reading, you could, with our eyes and our brain, work out what that actually meant and what it changed. The problem is when we're trying to analyse gene sequences, it's a little bit more difficult to do that because you have to be able to essentially replicate the normal in vivo situation. So all the things that we might do in a laboratory situation are in some way or other removed from what happens in real life. They're also very long, time-consuming experiments and very expensive experiments. So the way of the future here is partly improved computer-based analysis tools, and they have come along so far in the last 10 years, it's incredible, whereby we can actually ask a computer, what do you think it does? But whether that's actually the right answer, we're talking about a big decision here. If we've got a person who's sitting there saying, should I, shouldn't I have radical surgery, you want to be pretty sure that you're giving the right information. So again, it just comes back to making sure that the data that you have is actually reliable, and that's why it's hard, because you have to actually do this from lots of different angles and make sure that you're getting a consistent picture. Sometimes you do, sometimes it's straightforward and easy, and through the big consortiums where you've got lots of groups around the world all analysing the same variants, it makes it a little bit easier because you've got the power of numbers. But the other thing is the huge number of these variants that exist. So because they're rare, you can have 500 patients and they'll all have a different variant. So that makes it challenging as well.
Fantastic. All right, so we have a question here thanking you for a fantastic and a clear presentation, first of all, but asking if you have the BRCA2 condition, how does that manifest differently from BRCA1? Is there a difference?
So they're different genes, they're on different chromosomes. They both produce DNA repair proteins that have an incredible amount of similarity, but also a lot of differences. In terms of the characteristics of patients who inherit these diseases, I'm going to put a huge caveat in here, and that is that I'm not a medical doctor, I'm a scientist. So I am speaking as a scientist that I know that in the case of BRCA1, there is an increased risk of ovarian as well as breast cancer, whereas in the case of BRCA2, depending on the mutation, there is an increased risk of breast cancer and some other cancers as well, depending on the mutation. So similar genes, different characteristics of the disease.
Right. So question from Twitter, from Ron, who asks, how often does a normal breast cell divide?
It depends on the time of life. It depends on the cell division rate increases during puberty and it increases during lactation. an average human cell divides once every 24 hours, but it's incredibly variable depending on the cell. So some cells don't divide at all, other cells divide very quickly. It also depends on the situation. So for example, if we're talking about skin cells, they will perhaps divide every 24 hours, but if there's damage to the skin, they'll divide more frequently. It also depends on age.
Right. Couple of questions about breast cancer in men, and why is it less common in men, and also whether men are similarly affected by the BRCA1 and 2 genes.
So the frequency of breast cancer in women is about 1 in 11 in Australia. In men, I think it's about 1 in 100. BRCA2 mutations, but not BRCA1, are associated with an increased risk of breast cancer in men.
Great. Question here, where did I just put that there? So we've got a few questions around genetic testing and its implications. So some of the questions are broadly is, should we all be getting genetically tested for these and other mutations?
So anecdotal story, and that is 23andMe. I think it's 23andMe, one of the companies in the US where you could send your DNA by spitting into a tube and send it off, and they'd come back and tell you your disease risk profile. I remember at the time thinking, gosh, that's brave, because as I say, we don't understand, and we understood even less then. But, you know, there are, if you happen to have inherited, you know, that on that blue graph, if you happen to have inherited a defect in one of those very, very highly penetrant disease genes, then yes, that might be helpful to know. But once you start moving towards the left and you're talking about much less severe genes, and the fact that they're so dependent, one, on the other genes you've inherited, and two, on the environment, it's very hard to be confident in your predictions. Now, not long after that company, which I might have the wrong name, which is a bit risky, but they've stopped doing it, they're doing ancestry testing. Now, if any of you had a look at I think it was the Courier Mail a couple of weekends ago, my good friend and colleague Anne Trezise was interviewed about the fact that actually the genetic tests that are used in ancestry testing are also flawed, which was very disappointing because I was just about to go and have it done because I was having an argument with somebody about where I was from, but I'm not going to do that anymore.
And the last question, I don't know whether you know anything about, there's a few that have touched on it, is whether there are any insurance implications for genetic testing now and potentially in the future as we get more and more knowledge of this.
So that's where my question was coming from when I said, do we actually want to know and do we want others to know? Now, I don't actually know what the insurance policies say, and I'm not qualified to speak on that, but you know, it is an issue, and it's certainly an issue that's been discussed widely. So I think that's probably as much as I can say on this point, but it's a very, like all of the questions, very, very good question.
And I guess it shows the strong overlap between science and other areas of policy and ethics.
Absolutely, absolutely, which is why we have to do multidisciplinary research so we can work together and get these problems solved.
Yeah. Absolutely. Yeah. All right, well, I think we might, there are more questions here, but I would really encourage you to take up Melissa outside and ask those then. But I want to give you a chance to get to your food and also not have you under the lights for too much longer.
It's pretty hot up here.
So I'd like you to all please join me in thanking Melissa tonight for a fantastic presentation.
Thank you.
And we have a small token of our appreciation.
Oh, yay. Thank you very much. Thank you. Thanks very much.
I look forward to seeing you all next month, and please join us outside for some meal.